Hi guys. This is the third time I'm starting this because of all of the interruptions over the loudspeaker. So I'm hoping that we make it through, and if not, you might hear some announcements because I'm just going to power through. Chapter 3 is really quite intense. It is probably an entire year of biology stuck into one chapter. So I'm chunking it into 3 groups, this first group being cell structures and some cell physiology. You really want to focus on the physiology because most biology classes don't dive deep, and so we need to dive deep into how this stuff works. If you have not saved my playlist, consider doing that because I do add some videos to this, but there's way more on the playlist. All right, the first section here starts on page 95, with some beautiful pictures. And this is all about cells being the smallest and most fundamental unit of all biology. Typical cell is small, less than 1/10 of a millimeter in diameter. And you cannot be examined. We couldn't really see it until Robert Hooke, the 17th century. And then we started to really see some neat stuff. And we could come up with this cell theory. The idea that cells are the building blocks of all life. Cells come from pre-existing cells, and that they are the smallest, most fundamental units. This sort of describes a living organism. Now that we're going to talk about tissues, and that's coming up in the next chapter, but we need to start talking about those cells coming together and cooperating and differentiating. So each cell is able to maintain homeostasis, a certain level of cytoplasm, a certain chemical composition, a certain amount of enzymes, things like that. But also when they work together, they can coordinate this homeostasis, and that goes for tissues, organs, and organ systems. The bottom of this page on 95 here has this really nice picture that's coming up here about cell differentiation. So let's start at the beginning. An egg is fertilized by a sperm. That becomes a zygote. 2 cells become 4, 4 cells become 8, and it's this ball of cells, okay? These cells are not differentiated. That is, they have the potential to become any type of cell, a skin cell, a nerve cell, a stomach cell, whatever. They are raw, so to speak, okay? So it has this great amount of potential. Differentiation is when the ball of cells starts to get some regional differences. And what that means is there's kind of a head region and a tail region, so to speak. And then some of the layers of the embryo start to develop into have certain functions. And so they start to just differentiate. They start to have a certain function or a certain specialization. And that picture at the bottom of page 95 shows that, this once undifferentiated just keeps dividing and dividing and dividing until it reaches a point where some of the genes are turned on, and they can be turned on to become any of the 4 tissues of the human body. Moving on to the next section here, you can see that there's a nice flow chart and kind of pieces out all of the distribution of the cell and surrounding things. So basically everything outside the cell is extracellular and everything inside is intracellular. We biologists, we love all these different names. So in certain areas, the extracellular fluid can also be called interstitial fluid. And that's most tissues, but not all tissues. Some tissues have a special name for their extracellular fluid. Inside is called the intracellular fluid or cytosol. You probably heard of it called the cytoplasm, although it's not exactly the same thing. A plasma membrane is the barrier between extracellular and intracellular. The basic cell structure is that plasma membrane. Within it is a cytoplasm, which you can subdivide into the cytosol, which is the fluid part, which is probably what you call the cytoplasm in biology, and then the organelles, which are the little organs or the structures with certain functions, nucleus, mitochondria, endoplasmic reticulum, all that. Oops, went too far. Okay, so the next section is all about the cell and all of the organelles, okay? And so we can kind of subdivide it into those that have membranes and those that don't. And if you remember about membranes, they're made up of phospholipids, that bilayer of phospholipids. And we're going to talk about membranes here in a little bit again. But that's what we're talking about in terms of membrane. So the non-membranous, not completely enclosed by membranes, so they kind of are bathed in that cytosol. The membranous have that phospholipid membrane. A lot of them are part of the same system of making proteins and packaging proteins. And so they're isolated from the cytosol. And here you see some examples of non-membranous and membranous. The next several slides go through the different structures on page 97. So what I'd like you to do here is pause and really focus on the artwork on page 97. I'm going to actually skip through this because I want you to look at the structure, where it's located, and its function. And we'll come back in a bit. Okay, so we are skipping to module 3.3. There were some review videos also embedded in the slideshow, so if you didn't get a chance to watch those, you might want to watch those. We're going to focus on the plasma membrane. And the plasma membrane is a selectively permeable barrier. That means certain things can come in and certain things are not allowed to come in. And that has all to do with the molecular structure of the plasma membrane. So some things that it controls is the entry of ions and nutrients, the elimination of waste, and the release of secretions. Its composition, there's a nice picture on page 98, is of the phospholipid bilayer. It also has proteins, which can either be integral, transmembrane, peripheral, or the glycocalyx, which is a layer of superficial carbohydrates. And if you remember superficial from lab, you are on top of it. Okay, this is the picture of all those types of things. You can see an integral protein with this channel right here. Integral proteins, okay? You can also see peripheral proteins here. Those are the ones that are kind of just on, that's not one. Those are the kinds just on the edge. That's what peripheral means, just on the edge. Okay, but if it's integral, it goes through the whole membrane. It's integral, goes through the whole membrane. You can also see some cytoskeleton here, which also helps with the structure of the cell. You can see the phospholipids here, and the cholesterol is also indicated. So there's lots of stuff going on in the plasma membrane. The hydrophilic heads and the hydrophobic tails. The hydrophobic tails face each other. And then you have cholesterol interspersed. Cholesterol helps to maintain the integrity of the plasma membrane. It says stiffen, but it also keeps it very fluid-like instead of, a hard surface or layer. Integral and peripheral proteins. Integral usually are channels. I shouldn't say usually. Many of them are channels that can transport water solutes through the membrane. Peripheral proteins, they can kind of easily be removed, and they can have functions like to be an enzyme or to have some sort of role in a pathway, regulatory pathway. The glycocalyx, you got a lot of different molecules involved, the proteoglycans, glycoproteins, and the glycolipids. But what's more important is the functions. Some of these are like IDs, cell recognition. Some of them are able to bind to extracellular structures to make a very more rigid tissue. And some of them have lubrication functions. So this table here is showing you the functions of the proteins that would be found in the membrane. And then this is the left side here is the function of the membrane as a whole. So, not only does it provide that barrier, it's an isolation, but it regulates exchange. And that's really like the two big things. Excuse me. Pay attention to the different roles that, if you remember learning about proteins, they have just so many different classes and functions. But these are the functional classes within the membrane. So these are the ones that are important in the membrane. Let's go on to the next section, was about the cytoskeleton. The cytoskeleton, it's just kind of like it sounds, the cell skeleton. It provides an internal framework. And it gives the cytoplasm some strength, but also some flexibility. Some of these cells have to be odd shapes. So the cytoskeleton helps with that. And it's composed of microfilaments, intermediate filaments, and microtubules. Microfilaments are composed of the protein actin. They're commonly found at the periphery of cells. And there's a nice table of this, by the way, on page 101. And they're the smallest element. That's why they're called microfilaments. Some of the types, microvilli are like finger-like extensions. And a lot of times they are for increased surface area. So if you have increased surface area, there's more surface area for absorption to occur. So a lot of things like in your intestines might have some microvilli. A terminal web, this kind of is an example of microfilaments within the plasma membrane in cells, and they form like a layer. Intermediate filaments, these are the strongest and most durable. You can see some examples right here. And then microtubules, these are just kind of like they sound, they're hollow tubes, and they're built from tubulin. These are the largest component of the cytoskeleton, and they do play a part in cell division. So there's some that extend outward from the centrosome. So in the centrosome, you've got centrioles, and you probably talked about centrioles when you talked about cell division. They are composed of microtubules, a specific composition, 9 groups of triplets. And they control the movement of chromosomes during, during cell division, okay? And this is in animal cells. You might have remembered that plant cells don't have them because plant cells have a cell wall, so they don't need this more support. But animal cells do, so centrioles are critical. Cells without centrioles cannot divide. For example, red blood cells. Red blood cells do not divide. Also, skeletal muscle cells do not divide. That doesn't mean to say they weren't made from cells. They were at one point dividing, but then they go through a life cycle and they hit a point where they don't have centrioles anymore, so they can't divide. Red blood cells also do not have a nucleus. Cilia. Cilia are extensions of the plasma membrane. They're like short, hair-like, and most of the time they're for motility. For example, in your respiratory tract, you have a lot of cilia that line the respiratory tube because you don't want gunk in the very tiny little alveoli of your lungs. So the cilia help to trap. They also produce mucus, so they help to trap all the gunk. And then you like hack it up and you either swallow it or you cough it out. Modal cilia beat rhythmically. So these are, they actually beat in a motion which is regulated and coordinated, rather not regulated, coordinated, so that they beat the cilia in a synchronized way to get, and for example, in the respiratory tract, to move that stuff out of the lungs. Primary cilium, that is a sensor, versus flagella. They are longer than cilia and beat in a wave-like sensor fashion, and kind of guess there's really only one kind of human cell that has a flagella, sperm cell. And here is a nice table to summarize all this information. Some of the more physiological structures, and when I say that, I mean they're responsible for more, they have more function in the cells than others, are going to be on these following slides. So ribosomes, if you remember ribosomes for transcription and translation, ribosomes are composed of 2 subunits, one big one, one small one, and they have to join together before that mRNA sits right here and starts to translate. some are free throughout the cytoplasm, and some are actually attached to the rough endoplasmic reticulum. Remember, that's why it's called the rough endoplasmic reticulum. So that's where a lot of proteins are synthesized. The endoplasmic reticulum is a network of membranes. This is part of the membranous organelles. And they are basically a network, kind of like a maze. They remind me of a maze. Okay, some have ribosomes, some don't. The ones that don't, that's the smooth, and that's where a lot of your lipids are stored and synthesized and stored. And then the ones with the rough, that's where a lot of your proteins are synthesized and stored. Just go ahead and review that. So this is part of the endomembrane system. The ribosome is manufacturing these proteins. And before they're proteins, they're polypeptides. That's just the strand. Remember the different levels, primary, secondary, all that good stuff. So once they travel throughout the ER, they sort of gain more than just that sequence of amino acids as they become a fully fledged protein. And when they're ready, they bud off in these transport vesicles and they go to the Golgi apparatus. The Golgi apparatus helps modify these proteins, and if it's a secretion, they can get it into a secretory vesicle, or they can release it from the cell in exocytosis. And there's a great picture of this happening, page 105, 104, 105 actually. Transport vesicles arrive at the cis phase. So the system trans is just like the in and out of Golgi apparatus, the receiving side and the shipping side. The receiving side is where the transport vesicles fuse with the Golgi apparatus membranes. They modify the proteins, and then they're repackaged and they move towards the trans, which is where they are packaged finally and released as a secretory vesicle or some other vesicle. This is the picture from page 104, and this is a great picture with some great numbered steps. So take a look at that. Some of the products that the Golgi apparatus can produce, membrane renewal vesicles, that helps the plasma membrane change. So if we can make more of the proteins in the plasma membrane, we can, alter how the plasma membrane functions. Secretory vesicles, they can contain hormones or enzymes or, anything that extracellularly is needed. Lysosomes have digestive enzymes, and lysosomes do a lot of cleanup. So inside, if you have a damaged organelle, a lysosome will come and break down that organelle. Lysosomes, through basic functions: fusion with another organelle and digestion of the contents. That's what I was just saying. Fusion with another vesicle containing solid extracellular materials. So basically it fuses with it and it breaks it down more. Or it can release digestive enzymes, which help with autolysis, which is this like self-destruction. If the cell is injured or the cell is ready to go, that's how lysosomes help this process. And here's a great picture of the lysosomes. The Golgi apparatus membrane fluid this is a continuous shipping. Think of UPS, 24-hour UPS. Complete continuous system. Mitochondria. So mitochondria, this starts on page 106, are the powerhouses. So this is where your ATP is being generated. You need to review kind of the big picture of cellular respiration. And this is a good point here, that mitochondria, you don't have a fixed amount. If you need to produce ATP, that type of cell, like a muscle cell, has a lot, like in the heart, has a lot of mitochondria, versus red blood cells that don't really have to make ATP. They just basically have to pick up oxygen and carbon dioxide. Have none. They do contain a type of DNA called mitochondrial DNA. It is a double membrane. There's an inner and outer membrane. The inner membrane kind of folds, and they're called cristae. And then it encloses this liquid called a matrix. So certain parts of cell respiration are occurring in certain parts of the mitochondria. And here you see the cristae, which is the inner layer that folds. And here you see the matrix, which is inside. And here you see some very important enzymes that help to catalyze all these pathways. So cell respiration isn't just one pathway, it's multiple pathways. And there is a transmission electron microscope picture of a mitochondria. Times 50,000 times its original size. Okay, ATP production, this is respiration. There, if you remember, there are three big parts of that. Some teachers say four. Either way, those are the parts of the process. This is on page 107 that you see this. Glycolysis is the first stage. This occurs outside of the mitochondria, where you take one glucose that's 6 carbon long, and you break it up into 2 3-carbon pyruvates. Okay, so that has to happen before it can go into the mitochondria and do the rest of cell respiration. In the matrix, that inner, kind of like the cytoplasm of the mitochondria, the CO2 is removed from the pyruvate. So take off a CO2, it's one of the products. So the citric acid cycle removes the CO2 and the hydrogen atoms, and that is indicated in the kind of center there, and it is a cycle. It is a cycle. So enzymes and coenzymes use those hydrogen atoms to catalyze ATP from ADP. So that's where you're breaking up, you're taking the hydrogen atoms, the energy, and you're putting it into ATP. It also forms water as a byproduct. So this whole process took a big molecule, glucose, broke it into smaller molecules that the cell is more capable of using for things like muscle contraction or cilia movement or active transport, any of those things. They can't use glucose, they need something smaller. And there's the overview of respiration. I'm not interested in all the little mechanics of it, just you need to know the big picture, and this is a great picture. It shows you the big picture. Aerobic metabolism or cellular respiration. So if you want to go through that whole process, you need oxygen. So ATP production needs oxygen because of the mitochondria. Without oxygen, you can produce ATP, but it's a much, much smaller amount, okay? This produces about 95% of the ATP needed by the cell. There's other ways to make ATP, but it's much less efficient, and there's much less of it. That will do it for this section. On page 108, there's a nice review of the introduction to the cell and some cell parts and things like that. You're probably good on the cell parts, so look again at the physiology. What's happening inside of the cell? What do those cell parts do? And I'll see you in the second video.